T cells are cells of the immune system. They're cells that are involved in finding an infected cell and clearing the infection. What's been perhaps more interesting has been recently, in the past 10 or 15 years, we've had a real appreciation of the idea that T cells also are important in protecting us from cancer. Now we're starting to develop drugs which use these T cells and make them work a little bit better, and then those T cells will go and destroy the tumors, and patients will live longer. So we're really interested in this idea that the patient has these T cells in their body when they come into the clinic. The problem is that by the time that the patient comes into the clinic, they've had that tumor for a very long time, and so we don't really get to see what happened before. We don't see how it developed, we don't see how the T cells participated in the response when they were working, and then we don't see how it broke down. And so what my lab is doing is we're trying to understand how the T cells work and really what's broken about those T cells so we can try and fix them. We develop engineered models so that now the tumors will form from single cells, and ultimately they'll develop through all the stages that would naturally happen in the context of a human cancer. But now we can study what has happened in those early stages. So that's the first part, but that doesn't ultimately give us the T cells. So in order to get the T cells, we have to also engineer those models so that we put in something called a neoantigen. And neoantigens are just self-proteins that acquire mutations that allow the T cells to see them. And so that allows us to then study those T cells as they develop. We can then use techniques called immunofluorescence, which allow us to look where the cells are, who they're talking to. We use techniques called single-cell RNA-seq that allows us to understand transcriptomically how those cells are functioning, how they change as they go from, say, the lymph node to the tumor. And then we're also quite interested in broadly how the tissue changes, how they form these lymph node-like structures that are often seen in patients that contain thousands of immune cells, and we think they help to fight the cancer, but we don't really understand much about them. In the models, we can actually study how they're forming and how they're enabling the function of the immune cells. The goal would be to ultimately develop therapeutics that are based on the knowledge that we've learned from the models to try and figure out exactly what are the levers and switches that we need to flip to try and make these cells work when they're in a patient with cancer. Yale has two really good things going for it. The first of which is we have a really world-class set of physicians who have a long history of knowing how to get drugs that are helping patients into the clinic. The other thing is we have really good immunology research here, and that helps us to both do the research that's necessary to figure out what's wrong with the T cells, and then try and figure out how to fix them and put the drugs into the patient. In my lab, I'm really excited to watch the students that we're training go forth and now have careers where they work on how to fix T cells, how to solve problems in the clinic. So we really have both sides of the coin here at Yale.